little bit more prepared for your midterm. If you have not been going over those exams, checking to make sure what you got, and when you took it again with your notes, when you looked at it against the key, did you match up that score? Does that score match up what you got in Carmen's grades? If you haven't done those activities, then you are behind. So, you might say, but how can I be behind when I'm 4 weeks out from this assessment? Well, why not be very proactive? Because I am pretty sure that week before spring break, there are a lot of things on your calendar. This is one that is intentionally set up for you to be able to tick off early and not have to wait until the last minute to do it. Also, I can tell you from students' responses afterwards, that probably the biggest response I get in regards to the midterm is, wow, I really should have done that earlier prep and not tried to wait until the last minute to do it all. Trying to cram 7 exams worth of 7 times 15 questions into your head in that last week, it doesn't work for how a brain physiology happens. So that's why we have it set up week by week, small little bites. So make sure you're doing that. You can be happy when it comes 4 weeks from now. So jumping back into talking about our signaling, this idea in regards to our signal transduction, we saw our 2 groups that we can put our receptors into, which is based on our 2 categories of ligands. But now let's look at, well, what can we do to impact this signal activity? How can we change, upregulate or downregulate whether the ligand is able to bind to and activate a particular receptor? So let's start off with what can we do that can downregulate, decrease the likelihood of a particular receptor being activated. So the first thing we can do is, and let me state up front, remember we're focusing on the receiving cell. So that receiving cell actually can impact the ligand in this situation of wanting to decrease activation. Well, if that cell wants to decrease activation, what can you do to the ligand that's coming to it? Give you a hint. Right now, if you want to focus in on this class and not receive text messages, what do you do to not have those ligands come to you? You take this and you make sure that those ligands can't get through. Well, not as extreme because you want to get those ligands after class, right? So, a little bit more extreme in regards to what our cells do is not just use those ligands, but instead break them down, catabolize them. This is something that we especially see happen in regards to our neurotransmitters. As we get to move on to our nervous system shortly and see more about how our nervous system does our communication with neurotransmitters, we will see that one of the key parts is that yes, that sending cell releases the neurotransmitter, but our receiving cell actually destroys quite a bit of that. And we actually want it to do that process so that we can stop that communication from occurring, lower the amount of activation happening. But aside from changing what's going on with the ligand, another way that we can do it is change the receptor. So what can we do in regards to our receptor? Well, one thing is we could do the same thing. We could catabolize it. We could destroy it. So I say we do this when we see intracellular receptors, but we don't do it as much when we see membrane-bound receptors. And the big reason is that if we metabolize, destroy those receptors, and we want to bring that communication back online, what do we have to do? Rebuild those receptors, right? Would it be a really effective way for me to make sure that I'm focused on you guys and not on my phone by every morning taking this phone and throwing it against the wall? Did you think I was really going to let it go? Not that crazy. But that's what it would be, right? If I did that and then I had to buy a new phone every single Tuesday, Wednesday, and Thursday, right? That'd be incredibly costly. So, we have to do it in regards to our intracellular receptors. But let me talk about one more thing before we can see and answer. Why don't we have to do it for our membrane bound? And that is one other thing that we can do, aside from breaking down these receptors, is to change their affinity. Affinity is based on what two things? Affinity is based on what two things? Either one of them. Shape. What's the other one? Charge. So if we in some way are able to change the shape of that receptor, bind something to it, that'll change its shape. Change its charge. We're going to be able to adjust its affinity and change our activation ability. Now going back to our membrane-bound receptors, what's our solution for membrane-bound receptors that is not so costly as completely destroying our receptors? Where are our membrane-bound receptors? Name says it, they are bound in the membrane. Can our ligand go through the membrane? Thank you. No. You guys not pick up on what we were talking about yesterday or not want to talk today? I'm not sure which. Hopefully it's not want to talk today. So, we have a situation where when we're talking about a membrane-bound receptor, we know that ligand can't go across the membrane. So what do we know we can do to integral proteins in our plasma membrane? We can endocytose them to bring them in, exocytose them to put them on the surface. These receptors are that. So what would happen if we took our receptors and endocytosed them? They're now part of a vesicle inside the cell. How likely is it that that ligand is going to be able to cross the membrane and then cross the membrane again to get inside that vesicle to interact with that receptor? Slim to none, and Slim is on vacation, right? So, what we can do for our membrane-bound receptors is to endocytose them. Is there a cost associated with that? Yes. We know vesicle formation has a cost. But as far as changing back to, okay, we want to not have that communication happen. Okay, now we're at the point where we want to have that communication happen again. We haven't metabolized them. We don't have to remake them. We just have to exocytose them back to the surface. It's like taking that phone and putting it on mute. And then unmuting it once you want that communication to happen again. Much less expensive than throwing it against the wall and catabolizing it. Why do I have insulin up there? Because I'm pretty sure that a vast majority of you have heard that individuals can develop insulin resistance. Well, insulin is a compound associated with helping regulate our blood glucose, that figure behind your cells, it actually causes more of our gluts to be put on the surface of our cells so we get more intake of our glucose. Wow, we just put like 4 things that we've talked about this term together. Thing is, that if your cells already have a whole lot of glucose, they're like, I don't want to hear from insulin anymore. So the receptors that bind insulin get endocytosed. So now we have plenty of insulin coming and trying to tell this cell to pull in that glucose by adding those gluts. But there's nothing for it to bind to because we've endocytosed, brought inside those receptors. Those cells have become resistant to insulin. If we can exocytose those receptors for insulin back, we can get that communication happening again. In doing so, we've saved energy. By endocytosing our membrane bound, we don't have to remake them. We can just exocytose them back. We don't have that option for our intracellular, because if we put our intracellular inside of a vesicle, is our ligand going to be able to reach it? Can our ligand for intracellular receptors go across the membrane? Yes, that's how we get to intracellular receptors. So if we hide it behind another membrane, is that going to be effective? No. Endocytosis and then later exocytosing, not a valid option for our intracellular receptors. Now, when we're talking about our membrane-bound receptors, we saw that most of the time we have them as relays, more than just the ligand and the receptor, but things happening after the receptor. So that is another spot where our cell can go, all right, we can let the activation actually happen, but the consequence of that activation we can take care of by not allowing that channel to actually have ions flow through it, not allow that enzyme to actually be activated. So at some point later on in that relay, stopping the response process. Again, option for our membrane bound, not a situation for our intracellular. Now on the flip side, increasing activation, remember our focus is on our receiving cell. So our receiving cell has no control over the amount of ligand that's coming to it. As far as to increase activation, because the thing that we would want to do in terms of that would be have more ligand present. Well, that's determined by the sending cell. So we don't have any control over the ligand here. But we do have control over the receptors. So what can we do in regards to those receptors? Well, we can make more of them anabolize. That is something that we regularly do and we must do if we want to change the levels of our intracellular ones. We do that on occasion in regards to our membrane-bound ones. But more typically, how we regulate our membrane-bound ones is how do we get more membrane-bound receptors? How do we lower them? We endocytose and hit them. What do we do now to bring that communication back up to a higher level? Exocytose them, put those back into our plasma membrane. So we see that a lot happening in regards to our membrane-bound receptors. So we can sort of lower that, and then when we want it to come back, we just exocytose them back to the surface. Again, saving energy, because we know from week 1 that we have a limited amount of energy, so when we can do things that save energy like this, that's a big gain. And not having to remake these receptors is a big gain. So, our impacts we have in regards to what we can do, lowering or increasing our communication pathways. actually start looking at organ systems and not have to constantly be going, well, wait a second, we got to understand this. So wait a second, we got to understand that. So, you might say, well, you could do organ systems in any particular order. Sure. We could do them alphabetically, we could do them top to bottom in the body. I want to do them by importance. And also with this idea that we did all of this groundwork so that we can understand our organ systems, that we need to understand the organ system that we're going to need for all the rest of our organ systems. And that comes down to being our nervous system. So that is how we will walk through our organ systems as building on them or building up from them as the case may be. So, overview for our nervous system. Hopefully none of this is too shocking or amazing. Your nervous system is a communication system. Pretty straightforward there. As we talked about and defined the category of our neurons as one of our primary types of cells, they are defined by being able to do electrical activity. And this is a part of their communication capacity in that they utilize electrical activity to travel long distances in your body. We know distance is an issue because we like to do a lot of things based on diffusion, and what do we know diffusion has? A distance problem, exactly. So, we need ways to solve distance problems. Our nervous system solves a distance problem by, instead of using a signal that has to diffuse, to use an electrical message that is able to travel much farther distances. But you might say, well, wait a second, I thought they communicated by neurotransmitters. Yes, in the end, to do that cell-to-cell communication, the majority of the time it is by neurotransmitters, our signals, being our ligand that is released and goes to bind to a receptor on that receiving cell to get that message passed to the next cell. So putting those two things together to travel distance, we have electrical activity in that cell getting us the long distance, that final communication happening over that very short distance by diffusion with our neurotransmitters. So, one problem with this process is that the only cells that we can talk to are the cells that we're connected to. So if we want every cell in your body to know about a particular thing that's going on, your nervous system would need to be connected up to every single cell in your body. Spoiler alert, it's not. Because it is so hard to make and keep all those connections. So you can kind of think about it as sort of everybody in your contact list, you keep touch with them every single day. You keep touch with them every week. Right? It's really hard to keep making those direct connections. So we'll need to see how we can change that so that we can have some way to get around this limitation. Overall, the goal for our nervous system is to get as fast a communication as possible, and also to have it happen as quickly as possible, so if that information changes, our response can change quickly. If a saber-toothed tiger were to walk in that door right now, speed of recognizing, realizing, and getting activities to happen is a life or death situation. The faster it can happen, the more likely life is still going to be in existence tomorrow. But if it turns out that that saber-toothed tiger is just like purring and totally happy, well then we want to recognize that change and not run away from it. Maybe go, okay, it's not a life or death situation. Quick response changes. So we want things to be fast, but we want them to be of short duration so that that next message, which might be the exact opposite message, can get through as quickly as possible. Make sense? Alright, so, yes, we're doing all of this communication component, but the majority of that is so that our nervous system can be an area where we're doing a lot of processing, a lot of controlling of what the overall body does. Should sound very familiar. Not from outside of this class, but from inside of this class. And that we're seeing, processing, controlling, determining whether we have a response or not. That's all setting us up to see, this is a spot where we can use our reflex template and put our nervous system on top of that template. And things like our brain can be seen as being our integrator in our reflex template. So this system is going to be critical for us doing what we know is our most important piece of physiology for helping us with our homeostasis situation, helping make sure that these all thousands of parameters in your body are ideally all staying at steady state. And when one is not, how to get it back without throwing too many others out of steady state. Hence why this is the first organ system that we need to work with. So, as far as our template goes, and creating the equivalent of our reflex template for what is going on with our nervous system, we need to make sure that we at least understand a few parts in regards to naming-wise what our nervous system is. I'll tell you right now, leave a little bit of extra room in your notes, because we will not finish this template today. We will be adding to it over the next couple of days. So you'll move back and forth in your notes. So, first thing to mention is, I hope, it's nothing new to say the central nervous system. That at least you've heard that terminology before. And ideally, I would hope that some of you would be able to tell me what are the two parts of your central nervous system. Your brain and your spinal cord, exactly. Well, thinking about associating this with our reflex template, where would we put this in our reflex template? Right in the middle at that integrator spot. Right? I just said, integrator, we can think of that as our brain. It's determining, hey, getting inputs as to what your temperature is. Is it changing from 98.6? Do we need to do things to warm you up? Do we need to do things to cool you down? Or are we good for what we're doing right now? So, we can think about this as being that middle part on that linear version of our reflex template that we created. What that means that, well, what's the top part? What's the key component in that top part? What sends information to our integrator? Oh, little struggle there, huh? Right? We got a stimulus. Then we have our receptor sensors, afferent pathway to communicate. And then does our integrator actually get our response? No, it's got to talk with our effector. So we need a section of our nervous system that applies to that top part and that bottom part that are not our central nervous system. Our central nervous system is acting as that integrator. So those two other parts are our peripheral nervous system. So named because they are outside, they are peripheral to our central nervous system. Not named in regards to our reflex template, but named in regards to the organization pattern in regards to our nervous system. But for this, as I said, why do I have that double arrow up there? Because thinking about our reflex template, we got to be bringing information into our CNS from our PNS and then sending it back out. So, arrow in, top part of our template, arrow back to our peripheral nervous system, bottom part of our template. So we need two parts to this peripheral nervous system. We need an afferent part that is providing that sensory input. And we need an efferent part that is getting our body to do the responses that our central nervous system deems as necessary. So could we reorganize this so it matches our linear format of our reflex template? Sure. But as far as organization, what's actually happening, it's things coming in from our peripheral, things going back out to our peripheral. Hence, this is a better visual organization happening. But realize the two are actually the same. Good? So as I said, we're going to add some more to this, but I needed to make sure that we were all on the same page when I was talking about what the CNS is, the central nervous system, versus the PNS, the peripheral nervous system. Let me give a little editorial note. I know I need to put up the next figure set. So you don't currently have access to figure 36. You will after class. So, our basis that we need to first understand in regards to our nervous system is this critical cell, one of our primary types, our neuron. So here with our first figure 36, fortunately it's a nice, easy figure. We have our basic neuron, which I'm pretty sure to a person, if I said, draw me a neuron, you would have drawn something that looked like this. So what are the critical pieces and parts that we need to know of in regards to these neurons? Well, the first one is the cell body. The cell body in this picture is colored green and has that blue circle in it. What's the blue circle representing? The nucleus. So the cell body spot where we have the nucleus. What's that nucleus contain? That DNA, the genetic material. What do we do with that? We read it, we turn it into things like our proteins. So our cell body, one of the key things is, it has that nucleus, which is our instructions for being able to make key things like proteins. So this is also the spot where we see protein production. I mention that now because that's going to become very important when some of our neurotransmitters that are getting used by this neuron are actually proteins. They get made here in the cell body area. But what makes our neurons so visually distinctive from the rest of our cells isn't the cell body, which actually actually is far exaggerated in size in this picture. What we really notice and think about in regards to our neurons is what? All those things sticking off that green area. Those are called processes. Those are critical for this basic function of our nervous system, which is our neurons need to be able to do communication. So these structures coming off, 5 coming off the green arrow, a green area without any additional coloration, and the purple one coming down to your lower right on the figure, are these processes, 6 in this picture. But as you can see, and as I just explained them, there's 5 that look really similar, and then there's this oddball 1 that looks different. So, all of them, all 6 in this picture, are going to help us with this communication. They're either going to do input, gather information, or they're going to do output. They're going to send information on. But as I said, we can easily see that not all processes are the same. We definitely have 2 different groups to them. So, first group are the dendrites. These are the ones that are coming off of that green arrow, or green area, not arrow, green area, not the purple one. Right? You see no change in color reference for our 5 dendrites. So, thing is that this is a nice simplified picture of our neuron. Actual neurons in your body can have upwards of 400,000, just shy of 500,000 dendrites. Not something that we want to put into a basic picture of a neuron having that many things come off of it. So we typically simplify and show far less dendrites than are associated with a typical neuron. Here, 5. But I need you to realize that most neurons have far more than 5. Again, upwards of 400,000 of them. So, these all are distinctive because they are just coming off of our cell body. Distance-wise from that cell body, not very far, they're not very long. So they're not going to be doing anything too far away from that cell body. What are they going to be doing? Dendrites receive information. They are gathering. So the more dendrites we have, the more different cells this cell can be gathering information from. So those ones that have upwards of 400,000, that's like their contact list is huge. Because they can be getting information from upwards of 400,000 different cells. Now, why do we see them with sort of these wispy ends? Right? They sort of look like bad scarecrow hands. Well, because we want to have increased surface area. Because as we know from talking yesterday about our ligands to our receptors, that most of the time, our neurotransmitters that we're going to communicate via these dendrites are going to be in our polar category and bind to membrane-bound receptors. Well, if we have more membrane, we can have more receptors. Increased looks like a big tube coming off of there. So this is the axon, sometimes referred to as the nerve fiber. I don't like that terminology because it confuses things, and oftentimes when people think of nerves, we're talking about multiple cells. Here we're looking at just one cell. So, much prefer thinking of this as axon. Now, what do we have in terms of, we know distance is a component. We think of cells, well, cells are small things that we need microscopes most of the time to be able to see. And most of the time we can get a whole lot of cells onto one little microscope slide. So these axons must be pretty small because cells are small. Actually, axons can be this single elongated tube. Many of them, depending on the size of the individual, are over a meter in length. Wait. A part of a cell can be measured as over a meter? Over a meter? Yes. Your neurons are huge cells that go long distances. You have neurons that go from your spinal cord out to the tips of your fingers, down from your spinal cord at the base of your spinal cord out to the tips of your toes. Yes, through your shoulder, through your elbow, through your wrist. So every time you move those, you're putting force on this tube. A little bit cool, right? Not the way you thought of cells before. Yeah, you have cells, thousands of them, that run the entire length of your arm, the entire length of your leg. Not cells joined together in a train. A cell that goes that whole distance because of this axon being that long. So, it needs to go from our cell body, but it's not leaving our cell body in the same way dendrites do. It's doing it as this tube. Because we need to provide structure in that tube so that, for example, when you bend your elbow, you don't break this cell, break the middle of our axon, and now no longer have communication. We also need this to be a tube so that we can move things inside of that tube. So, we got a lot of structural components in here that make it be much bigger than our dendrites. Functions like moving things from our cell body out to the ends of our axons. We need a pathway inside of there, hence hollow tube. Structure, very important. How many of you, gosh, it's probably been about 6, 7, 10, I don't know. It's been quite a while. Anybody do the ice bucket challenge? Remember those? Why'd you do the ice bucket challenge? Because you got challenged. They said jump off the cliff, so you did. Does anybody remember what was the ice bucket challenge raising money for? Nice! ALS. You know what those letters stand for? Amelia trophic sclerosis. No, I'm not going to test you on that. It's about the fact that those individuals that have that disease, their axons can't stay as these elongated tubes. The structures inside them break down. Consequently, when they bend their elbow, they actually break a whole bunch of axons in that process. Changing the position of their wrist breaks axons. structure that's supposed to hold this tube together, there's a problem with it. So you actually knew a little bit of what we were talking about today, right? Alright, so it's going out from our cell body. Dendrites typically associated with gathering information. Our axon associated with sending information out. This ending part, which we can see, we got elongated tube, but then we get wispy ends again. This is referred to as the axon terminal. Name tells us everything. It's the axon's end terminal. So, not a difficult motive. Why is it all spread out? Well, so we can talk to more than one cell at a time. So that where all we have are these connections, as we send a message down this axon, every cell that we are touching with that axon terminal is going to get that message. So, axon terminals are branched. The more branching, the more different places that information can be spread onto and passed to. We're going to see this as the spot where we release neurotransmitters. Oh, wait a second. If those neurotransmitters happen to be proteins, wait, where were those proteins made? In the axon terminal? No, all the way back in that cell body, possibly a meter away from their release point. And again, being passed inside of that axon to get to the axon terminal where their ultimate release point is. Am I saying that all neurotransmitters are proteins? No. I'm just saying that the ones that are proteins, they got a long distance and they use that axon inside of it to get to the axon terminal. So, we got our cell body, we got our two types of processes, our dendrites, our axon. But I'm guessing a few of you are thinking, well, that was a reasonable picture of our neuron. I've seen that before, but I also know that a lot of our neurons are myelinated. So, why didn't she talk about another part of our cell, our neuron, that is myelin, that I've heard about? Well, because that myelin, as we can see, we've taken our base neuron and we got 3 pieces of myelin associated with it in this picture representation, indicated by the red pieces around our axon. I didn't talk about it as a part of our neuron because myelin is not a part of our neuron. The axon is a part of our neuron, but this myelin is actually other cells that are there to assist our neuron at being better at doing its job. Oh wait, which of our 4 categories are these cells? If they are assisting our neurons in doing their job, they're connective cells. Connective cells that are helping our neurons. They are doing this by extending their plasma membrane around that axon. So I think the mental picture that works well is if you've ever had an injury to one of your joints, you might have for a little while had to wrap it, right, in a band, one of those ace bandages, those long components, and you just kept wrapping it and wrapping it around. You guys are looking at me like you've never seen that happen, right? Well, that's what's happening in regards to these cells. They're basically sort of flattening themselves out and then wrapping and wrapping and wrapping themselves around this axon. So now, instead of that membrane from our helping cell. Then we wrap that around again, we got another 2 layers of that helping cell. When we wrap it around again, we got these multiple layers of plasma membrane surrounding our initial plasma membrane. We know our plasma membrane is a barrier. Well, we just amplified that barrier incredibly. So what this myelin does is provide insulation from the extracellular fluid. Another way to think about it is, many of you come into class with scarves on, wrapped around to keep your head and neck warm. Right? Layers, you're insulating yourself from that cold air that's out there. A little less today than the last couple of days because it's a little warmer, right? It's like a balmy above freezing today. Remember that acclimation component? So, insulation from our extracellular fluid. Well, we know these cells are connected cells, but can we be more specific about them? Sure. We actually have 2 types of cells that create myelin. So, all myelin, wrapping around, providing this insulation, but actually what cells do that job? 2 of them. What we have pictured here in this figure is a representation from our peripheral nervous system, PNS. Why we needed to know the distinction of PNS or CNS. So is this neuron in your spinal cord? Answer is no. This is outside of your spinal cord, outside of your brain. These cells are called Schwann cells. It's actually labeled in the picture. But this is 3 individual Schwann cells creating these 3 spots of myelin. And if we were to extend this axon, double it, we would need to have other Schwann cells come and participate to make that axon fully myelinated. So why do I have that 1 of 1 up there? Because every Schwann cell creates 1 section of myelin on 1 axon. So when we see this and we see, oh, we got no connections between these myelin sections, we must be looking at Schwann cells, which tells us we also know that we are looking at an area that's in the PNS, not the CNS. So right now, we've gotten a big clue as to when we start talking or seeing things set up in this way. We got a lot of location information that we can bring in without explicitly saying it. Because just by saying Schwann cells are present there, we know the configuration of how things are made up and the location where we are at. So if these make the myelin in our PNS, our other set of cells must make the myelin where? In our CNS. Those cells, still connective cells, still just doing this assistance for our neurons, are called oligodendrocytes. So named, and be careful, because we kind of have dendrite in the name, even though they are not associated with the dendrites, they are associated with our axon. Oligo means many. So these oligodendrocytes, as we can see from our figure 37 that I have up here, in the middle we can see the cell body of our oligodendrocyte with section coming down, including a myelin section over here. In this sort of tan coloration, we see this figure has 3 different axons, so 3 different neurons that are being helped. And we see creation of myelin by this one particular oligodendrocyte on all 3 of these. oligodendrocytes. Each one creates multiple sections, many myelinated sections of many neurons. I am not doing this, but let's just play the hypothetical case that I was willing to make a deal in regards to your grade. You need to give me 10,000 myelin-creating cells to get an A in this class. What are you going to hand me? 10,000 Schwann cells or 10,000 oligodendrocytes? How many say Schwann cells? How many say oligodendrocytes? How many say I'm not going to give you anything because I'm going to get an A all on my own? Why should the answer be Schwann cells? Because it's 1-to-1. How many spots of myelin are you going to lose? 10,000. How many spots of myelin are you going to lose if you give me 10,000 oligodendrocytes? 30,000, 40,000? Far more than just 10,000. Do you want to give me the least amount for your A or the most amount? The least amount, right? No, I am not doing that. Please don't say she's auctioning off grades or something like that. It's just an example. So, last thing to say today is, hopefully you've noticed that, well, wait a second, if this is providing insulation, it's not a very good insulator because if I think about other things that need to be insulated, like the electrical wires in my house, if there's breaks in that insulation, those wires are pretty bad. But we want those breaks. We need those breaks. They're so important that the individual who figured out the importance of these breaks, a guy by the name of Ranvier, even though most of the time we mess up his name and don't say it in French and call him the nose of Ranvier, they have a special name. Because it actually is going to be important for us to have periodic access to that extracellular fluid. We don't want to have continual myelin. So, tomorrow we'll start seeing why we need to have these nodes and getting into that area.